In this video, we'll carry on talking about blood vessels. And before we go too far, I'd like to talk about the concept of an aneurysm. And an aneurysm is a weak point in an artery or in the heart wall. And this often forms a thin-walled and bulging sac. And often this bulging sac will pulsate with each heartbeat. And of course, the big risk about them is that it may lead to eventual rupture. The term dissecting aneurysm, the blood accumulates between the tunics. Remember, arteries have layers, 3 different layers. So the blood will accumulate in the between the tunics of the artery, and it starts to separate them. It starts to basically get in there and wedge the layers apart, and that's why it's called, not surprisingly, a dissecting aneurysm. And usually we see this, especially with the tunica media. Now, where are some of the most common sites? The abdominal aorta, the renal arteries, the arterial circle at the base of the brain are some of the common sites, but certainly can happen elsewhere. Sometimes they can be asymptomatic, but sometimes they can be symptomatic and cause pain, especially if they're bulging and pushing against other tissues. And of course, if it ruptures, you can end up with a hemorrhage. And now, what causes this? There's a number of things that can do it. It could just be a congenital weakness of the blood vessel. So during your development or the patient's development, for some reason that part of the artery, they didn't lay down enough material or not the right combination, or there was some defect within it. It could be trauma to the blood vessel has weakened it, or it could be infections. And of course, 2 of the most common causes are atherosclerosis and high blood pressure or hypertension. Here we can just see an aneurysm here. Here's the abdominal aorta, and it should sort of kind of run in, should sort of more or less maintain the same width, but you can see the bulging here. And here we are right down near the pelvis where it's starting to bifurcate into the common iliacs. Now, within our arterial arteries, there are arterial sense organs, and these are structures that are in the walls, not every blood vessel, but many of them, many locations, and they typically are measuring 2 things. There's some for pressure and some for chemistry. And we've come across these because we have talked about baroreceptors and chemoreceptors in previous chapters. And of course, things like the carotid sinus, we have baroreceptors. It's in the wall of the internal carotid artery, and it monitors blood pressure and allows for basically what's called a baroreflex, which is a response to changes in pressure. We also have in the carotid bodies chemoreceptors, and we've talked about those, and they're associated with the common carotids, and they measure or monitor blood chemistry. And of course, they transmit their signals to the brainstem and the respiratory centers. And as we've talked about in previous chapters, they look at pH, CO2, and O2. We also have in the aortic bodies, we have their type of chemoreceptor. And there's 1 to 3 in the walls of the aortic arch. And it's basically the same structure and function as up here, but the difference is which nerve they innervate or they work with. And we can just see the locations here. We can see up here the carotid sinus and the baroreceptors. We can see the chemoreceptors also in the associated with the external carotid. And then as we move down, we can see here are baroreceptors in the arch of the aorta or the aortic arch, and we also can see that we have some aortic bodies also associated with that arch. Now, when we're looking at capillaries are where we do exchange and gases, nutrients, waste, hormones. And what are they made of? Well, they're thin-walled, and we're really looking at endothelium and some basal lamina associated with it. Now, there are some places in the body where we really don't see capillary beds, and tendons, ligaments, epithelia, cornea, and lens of the eye. Now, when I look at capillaries, there's three main types. There's the continuous, the fenestrated, and the sinusoids. And they all have slightly different structure and different functions. The continuous ones are the ones that you see in most tissues. It's the most common. And they're endothelial cells that have tight junctions. But they do allow the passage of solutes such as glucose. Now, what we also see are pericytes that wrap around the capillaries and contain the same contractile protein as muscle. And this can help to regulate blood flow and cause contraction and relaxation of parts of these vessels. Here's just a cross-section of a continuous capillary. And we can see that here is one of the endothelial cells, another endothelial cell, and a third endothelial cell. And you can see that we do have little intercellular clefts. Now, of course, they're too small for things like red blood cells or many big proteins, etc., to get through there. But water and other things can get through there. Here's one of the pericytes that we see associated with the capillaries. That's the body of it. And we can see a red blood cell within here. You can also see that there's other transport mechanisms that you don't just rely upon these clefts for things to move in and out of the capillaries, is that we can also have penocytotic vesicles where we can actually bring stuff in through that mechanism. And you can also do an endo and exocytosis or transcytosis across some of these cells. You can see here that often in between the cells we have tight junctions. Now, another type are fenestrated. And fenestrated, think of it as having little windows. And these are capillaries that show up in places where I need very rapid absorption and filtration. So the kidneys and the small intestine. And these are endothelial cells that have a lot of these filtration pores within them. They're technically known as fenestrations. And there's a very little thin glycoprotein layer. Now, what's the advantage of having these fenestrations? It allows for the passage of more things than a continuous capillary, but we're not everything. We're talking fairly small molecules. So that ensures that things like proteins and larger particles will stay in the blood, but certainly more things can come and go versus the continuous capillaries. And we can see here continuous capillaries. We have intercellular clefts. We have the fenestrations. So you can see there's fenestrations, pores, or the filtration pores if you want to see them. And you can actually look over here, and this is a surface view. This is looking down on the surface with electron microscope, and you can see the individual filtration pores. So it really does have a completely different architecture because it's serving a different function. Okay, let's go to the third type, which are sinusoids. And they're found in the liver, the bone marrow, and the spleen. And these are basically irregular blood-filled spaces with large fenestrations. That these fenestrations or pores or openings are large enough to allow proteins to cross. And of course, one of the big proteins we were looking at when we look at blood is one called albumin. Clotting factors, new blood cells can enter the circulation through these sinusoids. And here we can see a sinusoid. Now, you can see that here's the endothelial cells of the sinusoid, and you can see that the space in between is very large, so large that you can actually see red blood cells can work their way between them. So wherever I see sinusoids, it's really about large molecules, blood cells being able to get from one side to the other. So this is an example of sinusoid in the liver. And we're going to end it there, I think. We'll come back and talk about capillary beds in the next video.